Welcome to the biology lab video on the scientific method. The scientific method became widespread most famously in Europe in the 1600s and 1700s, when biologists such as Anton von Leeuwenhoek and physicists such as Isaac Newton joined many others in an attempt to understand and make order of the natural way of science. Scientific method sometimes is presented as a rigid set of steps to be followed, but its goal is not simply to follow those steps. The goal of a working scientist is to understand something about the natural world. Understanding in science comes about through observing nature, coming up with an idea of why nature is behaving the way it's observed, testing the idea, and then having other people test it before a conclusion is reached. So the first thing we do in science is make an observation. So you can observe those candles there, and we'll come back to those in a second. You're making an observation as you do that. Once an observation is made, we do some experiments. Sometimes the experiments are going to support the proposed idea, then we call it a hypothesis, and sometimes they may lead to more questions. So after we have an observation, we come up with a hypothesis, and then we do an experiment. In science, the old saying is definitely true. The more we learn, the more we realize we have still to learn. That's why scientists maintain labs and do experiments. There's so much to learn. Take, for example, the study of ulcers. As recently as the 1980s, virtually all doctors were fairly certain that ulcers were caused by stress and spicy foods. In the early 80s, when two Austrian doctors proposed that ulcers were in fact caused by bacterial infection, no one believed them. So in 1982, one of the physicians drank a beaker full of Helicobacter pylori culture, and as a result, he developed ulcers. After extensive testing around the world, it was found that ulcers are indeed caused by bacteria and are now treated. The scientists in that case made an observation about ulcers. They developed a hypothesis about the bacterial infection. They found the bacteria in the stomach, but nobody believed that that was the cause of ulcers. So one of the doctors did an experiment. He drank a culture of the bacteria. Now, after he did that, that was not the only experiment that was done. Other people, to my knowledge, didn't drink cultures of bacteria, but they did do more testing on ulcers. And it was found that, well, let me go over here. This bacteria, right here, found in this brown stained bacteria, this picture is from Wikipedia, found in the stomach tissue was indeed the cause of ulcers. So after extensive testing, they were able to determine that. It was repeatable, and they were then able to reach a conclusion that Helicobacter pylori was indeed the cause of ulcers. Ulcers are now treated mainly by antibiotics. These scientists were awarded the Nobel Prize in Medicine in 2005 for their work. So they were able to reach that conclusion, and now doctors use that information. The scientific method is actually something we each use every day. For example, if you go into a room and flip a light switch and the light doesn't go on, you may hypothesize that the light bulb is burned out. You may hypothesize the power's out. Then you would design an experiment, such as changing the light bulb and seeing if that has an effect, or checking another appliance to see if the power was out. That would allow you to reach a conclusion about that hypothesis. So we're going to do a little experiment here to demonstrate. On the screen you have two burning candles. So think for a bit about what these candles need in order to continue burning. When you do that, you're making an observation. So when you think about these candles, you probably come up with things like oxygen, wax, a wick. Without these things, a candle wouldn't continue to burn. Coming up with this, a candle needs a wick to burn, a candle needs wax to burn. Those are hypotheses. We can develop next an experiment to test our hypotheses. If we take away one of the components, let's say we're going to take away oxygen, the candle will no longer be able to burn. By coming up with that, that's a hypothesis. So we could do the experiment. So let's take away the oxygen from the pink candle here. We'll put a mason jar over it. As you watch that candle, if our hypothesis is correct, then by putting the mason jar over the candle, eventually the candle will use up the oxygen and will no longer continue to burn. As you watch it, you can see the flame is getting smaller, the candle is using up the oxygen, there's no way for more oxygen to get into the mason jar, and so as the oxygen is used up by the burning candle, the flame does go out. Not having oxygen prevents the ability for that flame to continue. To be a good experiment, this must be repeatable. So it's not enough to do just an experiment just once, but we're not going to keep doing the same experiment. If we did, in fact, light and cover the candle again, it would go out again. In fact, I've done this experiment dozens of times with these same candles, and they've always gone out. So we can reach a conclusion. Candles burned in the absence of oxygen will no longer continue to burn. So that would support our hypothesis. A final thing to notice about this experiment, I used 2 candles, not just one. Why was that? If you said control group, you are right. In order for us to be certain that the experiment was the cause of the effect, in this case the cause of the candle going out, we must have another group that is not exposed to the experimental variable. If we did not have a control group, which in this case is the purple candle, we would not, our experiment would not be repeatable. It wouldn't be scientifically valid. In order to be a scientifically valid experiment, we have to have a control group to compare our results to, and we have to be able to repeat the experiment. As we go through the course, you'll be performing some experiments. Experiments are not done always to reach some arbitrary answer. In this class, experiments are conducted to learn the process of science. So, for example, when you do your plan experiment over the next few weeks, don't panic if everything doesn't go exactly as you anticipate. That's the fun of science. Enjoy the journey and learn along the way.